Before I begin, I just want to acknowledge NSERC that funded this work and a very talented PhD student, Corey Bamel, who just finished and defended his thesis from the University of British Columbia. And so he was the one that did all the experiments in the work that I'm going to talk to you about. So my lab is very interested in mesolimbic dopamine neurons. And there's, over the years, there's been a lot of data showing that these neurons really respond to cues that predict rewards. And so it's been shown that these neurons fire in tonic-like or low-frequency firing-like activity, but when a cue that predicts a reward is present, that these will switch their firing to a phasic or burst-like firing activity that ultimately delivers increased dopamine concentrations to target regions, and that change leads to a change in the animal's behavior. For example, they might drug seek or food seek. And of course, that idea was based on these experiments done in Wolfram Schultz's lab almost 20 years ago, showing that when there's an unpredicted reward, you get this increase in phasic firing of these mesolimbic dopamine neurons. However, as the animals learn that a conditioned stimulus or cue predicts this reward, you get firing now to the presentation of the cue. And then when the animal is expecting a reward to be delivered but doesn't get it, you get a pause in firing. And they came up with this idea that dopamine neurons encode a prediction error. Of course, this was very much the dominant idea in the field, but now we know so many more things about dopamine neurons, and in fact, they encode effort, they encode incentive salience, they also encode aversive events. And it's been proposed that you have different populations of mesolimbic dopamine neurons that encode both positive and negative salience. So the model of these dopamine neurons is a homogeneous population that is involved in reward has really changed. And so we know that the outputs of these dopamine neurons vary. For example, you have projections to different regions in the nucleus accumbens, projections to the basolateral amygdala, and projections to the medial prefrontal cortex, amongst some of the major projections. And they also are very heterogeneous in their inputs, in that they receive inputs from the PPTG, the lateral dorsal tegmentum, the locus coeruleus, BNST, GABAergic input from the ventral pallidum. So just to name a few. So their inputs and outputs are incredibly diverse. The neural population, that there's dopamine neurons and GABAergic neurons and glutamatergic neurons, also makes them diverse. And the fact that they can actually release both dopamine and glutamate, and in some cases dopamine and GABA, also changes the function of these dopamine neurons. So, this is some work done by Stefan Lemel, who started this work in Jochen Roper's lab and continued it in Rob Malenka's lab. And he was looking at the electrophysiological characteristics of these dopamine neurons with knowledge of their projection targets. So he found that the mesolimbic dopamine neurons that project to the lateral shell exhibit a current called an IH current, or hyperpolarizing current. And they respond to in rewarding events. For example, when you do a conditioned place preference, you'll have activation of these neurons. But they also respond to aversive events. And then mesolimbic neurons project to the lateral shell. So these are both nucleus accumbens projecting neurons, just to different regions. They have less IH, and they very much respond to rewarding events, whereas mesocortical neurons that project to the medial frontal cortex seem to be primarily involved in aversive events. So then he went on to look at the different kind of inputs going to these different dopamine neurons and the projection target outputs. And he showed that dopamine neurons that receive input from the lateral dorsal tegmentum primarily project to the lateral shell and primarily are encoding rewarding events, whereas dopamine neurons that receive input from the lateral habenula, either directly or indirectly, primarily project to the medial prefrontal cortex, and that is thought to mediate some of the encoding some of the aversive events. So the point of all these slides is just to show you that dopamine neurons are really diverse in their function based, one, on their inputs and outputs. And so the question I'm really interested in is how are these dopamine neurons neuromodulated? So my lab's been very interested in this peptide called Orexin. It's also called hypocretin. And it seems to be important for motivated events. So you get increase in firing of these orexin neurons during reward seeking and mainly during wakeful states. And so it's thought that, and you also get increase in firing during stressful events as well as during sexual opportunities. And so it's thought that orexin neurons might transform motivational action into adaptive behavior. So they seem to be really involved in this behavioral activation. But so again, these neurons are located in the lateral hypothalamus, and they make widespread projections throughout the brain. And they do make a fairly dense projection to the VTA. And it seems like it's primarily an en passant projection up right to the locus coeruleus. But the VTA has quite dense orexin receptor expression. So to complicate this story, 95% of orexin neurons are co-localized with dynorphin. So dynorphin is an inhibitory, or I should say it's another neuropeptide, an opioid neuropeptide that activates inhibitory kappa opioid receptors. And whereas orexin activates GQ-coupled orexin receptors. So it's very interesting why an excitatory peptide, or a peptide with excitatory actions, and a peptide with inhibitory actions are co-localized in the majority of these neurons. And it's not just the co-localization within the neuron itself. They're actually located in the same dense core vesicles. So this is work done from Bill Carlson and John Muschamp's lab that we collaborated on. And with a similar distribution as what I showed you from Tom Joe's work, that you see co-localization. So the big gold particles here are dynorphin, and the smaller ones are orexin. You see a similar co-localization right in dense core vesicles. And so this is in the lateral hypothalamus, but they saw that these dense core vesicles containing both peptides are located in the cell bodies, dendrites, and axons. So presumably, these peptides are released together. And so likely they're going to neuromodulate circuits together at the same time when they're released. So they went on to do some behavioral experiments to show that actually orexin and dynorphin are acting in concert when the animal's engaged in reward-seeking behavior. So you can block the reward cocaine-seeking behavior when you administer an orexin receptor antagonist. However, when administering kappa receptor antagonist, the animals continue to seek cocaine. So we were really interested in whether or not orexin and dynorphin might act in the VTA to modulate different circuits. So it's the reason why you have an excitatory peptide and an inhibitory peptide being released together is actually to tune a circuit through different projections. So we used retrograde beads, fluorescent beads, from the lateral accumbens shell and the basolateral amygdala to test this idea that orexin and dynorphin might modulate circuits in different ways. So we waited for 2 to 3 weeks to get retrograde expression of the fluorescent beads in the VTA. And then we cut brain slices and recorded from midbrain dopamine neurons. Excuse me. So to identify dopamine neurons, we use these PIDX3 GFP mice. So PIDX3 is a transcription factor that's involved in the development of dopamine neurons. And you get GFP fluorescence selectively in mesolimbic dopamine neurons. And so it was a very easy way to mark the presence of the beads in these dopamine cells. So in the first experiment, we were looking to see whether or not nucleus accumbens shell or basolateral amygdala projecting dopamine neurons were overlapping populations. So in the same mouse, we administered a red fluorescent dye in the accumbens shell and a green fluorescent dye in the BLA, and looked for the overlapping dopamine neurons in the ventral tegmental area. Interestingly, most of these beads were located mostly in dopamine neurons. So only a few non-dopaminergic neurons had the beads in, but these were mainly dopaminergic projections from the VTA that went to the nucleus accumbens or the basolateral amygdala. Similarly, the density of these projections were pretty much the same. However, these projections were non-overlapping, suggesting that the red beads from the nucleus accumbens shell and the green beads from the basolateral amygdala were in separate populations of dopamine neurons. And you can sort of tell from this image, it seems a little blurry to me, but they're actually in regionally distinct areas with the basolateral amygdala projecting dopamine neurons located a bit more medially. So next, we wanted to test if they had different electrophysiology properties. We looked at IH current. So this is a hyperpolarizing cation current. And traditionally, it's been used as an electrophysiological marker for dopamine neurons. However, it's less, many dopamine neurons don't actually express much IH neurons, and it's IH current, and it seems to be more associated with the neurons that project to the lateral shell of the nucleus accumbens. And you can see here that the IH current in the lateral shell of the nucleus accumbens is much bigger than that in the basolateral amygdala. Similarly, the leak current, which we measure right about there, is also bigger in accumbens shell projecting neurons compared to the basolateral amygdala neurons. Looking at some action potential properties, we found the threshold to fire is no different between the two cells, but the spike width is bigger in basolateral amygdala. The spike height was smaller in basolateral amygdala, and the AHP minimum and AHP was bigger, but the AHP width was smaller. So it decreased AHP after hyperpolarization potential. So this part of the action potential can be associated with an increase in firing rate or an increase in excitability of these cells. So we kind of used an indirect and a direct measure to look at the relative excitability of these cells. And this was looking at how much current you have to inject to get the cell to fire for action potentials. And we found that in BLA neurons, you inject much less current to get the cell to fire for action potentials. So suggesting that they might be at a more depolarized state already, or in a more excitable state. And consistent with that, they had a higher firing rate than accumbens shell projecting dopamine neurons. So the next question was, how does orexin modulate these two populations? And so to make sure that we were going to activate use a saturating concentration of orexin to activate the majority or cause the biggest effect on firing rate. We did a dose response curve, and we found that 100 nanomolars was a saturating dose. So with 100 nanomolar, we should be able to activate the majority of receptors on dopamine neurons. So then we looked at first on accumbent shell projecting dopamine neurons. And so the blue neurons, or the blue circles here, represent the effect of orexin on the evoked firing rate, and you can see that there's a big increase in firing rate. Previous work that I've done has shown that orexin potentiates glutamatergic synaptic transmission. So one of the things we're wondering is orexin increasing glutamate to increase the firing rate of these cells? But when we applied synaptic blockers, there was no change in the firing rate potentiated by orexin. So suggesting that orexin might be inducing some sort of intrinsic change to the neuron to make them fire faster. And this effect was mediated by orexin-1 receptors, as it was blocked in the presence of the orexin-1 receptor antagonist. So looking at the BLA projection, there was no effect of orexin on firing in this projection of cells. So, and this was in the majority, or 90% of the neurons. So we found this really interesting, that orexin is selectively activating accumbens projecting neurons and not activating BLA projecting neurons. So then looking at dynorphin, again, we did a dose response curve to make sure that we were using a saturating dose, and 100 nanomolar was a saturating dose that gave us a maximal effect. And unlike orexin, dynorphin's effects were less easy to discriminate based on projection target. So so this is looking at accumbent shell projecting neurons. And the purple circles here represent the average of all the responses together. And you can see here, when you average everything together, there is just a little inhibition. However, when you divide them into responders and non-responders, you find there's definitely a population of kappa opioid sensitive neurons that mediate an inhibition of firing in this pathway, whereas the majority of these neurons don't respond. So we haven't really characterized further yet, but it'll be interesting to look at what other properties are associated with these neurons that are actually responding to dynorphin. So looking at dopamine neurons that project the basolateral amygdala, again, the purple circles here are the average response altogether, and you can see a much stronger inhibition. And that is because the majority of these neurons are actually inhibited by dynorphin, whereas a small population are non-responders. And again, just to confirm that this is mediated through kappa opioid receptors, we were able to block the response with a kappa antagonist, or BNI. Because dynorphin also can activate mu opioid receptors, which are present on dopamine neurons, or a population of dopamine neurons. So just to summarize so far, BLA projecting dopamine neurons are more excitable than accumbent shell projecting dopamine neurons. Orexin increases firing in the majority of these accumbent shell projecting dopamine neurons, but does not modulate firing in the neurons that project to the basolateral amygdala, whereas dynorphin inhibits a small population of accumbent shell projecting neurons, but then inhibits the majority of the BLA projecting neurons. So likely when you get release from these orexin/dynorphin neurons from the lateral hypothalamus, when you have release into the VTA, it's likely tuning circuits and biasing it so that you get increased dopamine release into the nucleus accumbens as opposed to the basolateral amygdala, which might drive more reward-seeking behavior. With that, I want to thank Corey Bamel, who did the majority of this work, and my funding sources, Brain Canada, that supported Corey Bamel's studentship, and NSERC, that supported this project. And thanks for your attention.
Thank you. Fascinating. I'll take a question up here. I guess there's a question in the front.
Hi, thanks for the talk. I love the story about how multiple neurotransmitters can get released from a single population to tune the sort of output. That seems like it could be put to use in other brain areas too. Are there other examples of multiple neurotransmitters being released into a heterogeneous pool of output?
Sure, yeah, like for example in the hypothalamus, like AGRP is co-localized with NPY. Most neurons have multiple neuropeptides that are located, and it's really unknown if they're actually located within the same dense core vesicles. I think the Orexin Dynorphin system is fairly unique in one. It's these peptides are expressed or synthesized in a population, a very small population of about 4,000 neurons that project like pretty much everywhere throughout the brain, and that those peptides are actually present in the same dense core vesicles, even though they have different precursor proteins. So I think it's kind of a unique system, but the VTA is not the only place where these peptides are released. They're released in multiple other brain regions. So I mean, I think it'd be fascinating to look if they're involved in tuning other areas. Hey.
So I try to follow up the question about co-release of the two peptides. But on the other hand, you also mentioned that the two populations of neurons that project to different sites are spatially sort of segregated.
Yeah.
So try to reconcile these two together. You see a differential effect, but on the other hand, they are co-released. The question is whether these two populations express different receptors for these two peptides.
These two peptides expressed.
Because otherwise, it's a little inconsistent with co-release, but they are spatially
distinct, whether you get a sufficient volume transmission and stuff. That is a good question. So like, for example, if the en passant fibers are closer to the nucleus accumbens projecting cells versus the BLA projecting cells, I mean, I guess that we don't really know. Like the only thing that I could say in support of this is that it's a fairly dense en passant projection through the VTA. And I'm making a point that it's en passant in that it's not synaptic release of these peptides. In fact, there's about 5% of the synapses are synapses from these orexin neurons. But they probably are volume transmitting to a certain extent, but you're right, like it's a broader population. We've only looked at 2 projection targets, and we plan to add to that. But it's a good question, and that I don't have a solid answer to yet. Yeah.
I'm sorry if I missed this. Since NorBNI can have some very long-lasting effects sometimes, I'm curious if you've looked at the effect of NorBNI on uncovering additional effects of orexin agonists. Yeah. So in that paper by John Mouchamp, one of the questions was, is there influence on NorBNI on orexin receptor signaling and that kind of thing, and that's influencing the behavior? And I think they did a bunch of experiments to address that and suggesting that they were signaling separately. But I think, At the population level rather than an individual level.
Yeah, so I don't know the answer to that. I mean, these were just in this is a bath application and not an in vivo experiment. So the time frame was fairly short. But yeah, I mean that's a question. Like you know, in retrospect, after we've done all this work, it would have been really nice to like suck up the cell and do RT-PCR and get some kind of characterization of what other mRNA is present for what receptors and stuff to see if we can separately identify these cells. But the other issue is that you know, the presence of the mRNA doesn't necessarily predict the activity of the receptor at the cell membrane and stuff, since that's on its own is a different level of regulation. But yeah.
Okay, question in the back.
Hey, thank you for the talk. Hopefully I'm on the right page here, but I think you mentioned that Orexin increases neuron firing rate, and I'm just wondering if it's more like Orexin allows the neurons to fire more, or they're making them fire more, and if you had a hypothesis on why that is so.
Yeah, so we're looking into the mechanism. I think it's a direct effect on the intrinsic properties of the neuron. We don't know what ion channel the signaling pathway is influencing, but we're looking specifically at that at the moment.
Okay, well, thank you very much.